Assalamu alaikum dear student, welcome back to another class with me. I'm Dr. Sajjad. In today's class we will explore the fascinating world of bioinformatics, which is a field, emerging field which merge science and technology, in an incredible way. So if you are curious about that how scientists use computer to unlock the secret of life, if you are a student, if you are a researcher, or if you are someone who just loves science, so this video is for you. In this class, in this video, we will discover what bioinformatics really is, okay, and how it's work with other related fields. So let's get started on this exciting journey into the world of bioinformatics. This class is for base student, this class is for master student, and this class is for researcher. So dear student, as I mentioned before that today topic is bioinformatics. So let's start with a basic question. What is bioinformatics? Bioinformatics is a multidisciplinary field that combines biology, computer science, mathematics, and statistics to analyze and interpret biological data. If we see the word bioinformatics, we can break down the word into many parts. For example, bio, infor, may, TICS. Bio mean for biology, N for mean information and technology, MM mean maths, TI mean text, statistic, and CS mean computer science. So it is a branch of science that integrate computer science, mathematics, statistics, chemistry, and engineering. Usually, we divide the bioinformatics word into two parts: bio and informatics. Bio mean biology and informatics mean refer to the data management and computational processes. So bioinformatics essentially means managing and analyzing biological data using computational tools. Bioinformatics is not a single field. It easily connects with other areas like computer science, engineering, chemistry, math, statistics, biology, biochemistry, genetics, medicine, and even in agriculture. For example, bioinformatics help us understand how genes work, find mutation that causes some diseases in human, animals, and plants, and create personalized medicine based on these informations. It helps us break down complex biological data and turn it into a useful information. Now the question, why we use bioinformatics? Well, in biology, we generating a huge amount of data. For example, if you take the DNA sequencing, RNA profiling, and protein interaction, so these data it will produce huge amount of data, and handling this huge amount of data manually, it is nearly impossible to handle it manually. Therefore, bioinformatics role came into science, and it allows us to store data, it allows us to retrieve data, and it allows us to analyze data and interpret biological data efficiently using specialized software. So why we use bioinformatics? We are using bioinformatics for analysis of data, for exploration of data, for integration of data, for exploitation of biological data in research and development. So these are the reason by which why we use bioinformatics in these scientific fields. to analyze data, to explore data, to integrate data, and to exploit data. And what type of data it will be? It will be a biological science data, which will be used in research and development. Now, bioinformatics deal with storage, retrieval, analysis, and interpretation of biological data using computer-based software and tools. Now, what is data storage? So bioinformatics help us store a huge amount of biological data. What will be the data? The data will be DNA sequences, RNA structure, protein sequences, and it will organize it in accessible way, and it will organize and store it in databases. Okay, these databases include gene bank, EMBL and UniProt etc. अब बायोइन्फॉर्मेटिक्स को हम किस लिए यूज़ करते हैं? बायोइन्फॉर्मेटिक्स को हम इसलिए यूज़ करते हैं कि इसके ज़रिए हम डेटा को स्टोर कर सकें, डेटा को रिट्रीव कर सके। रिट्रीव मीन कि उस डेटा को हम दोबारा यूज़ कर सके। जो डेटा हमने सेव किया है, स्टोर किया है, उस डेटा को हम दोबारा सेव यूज़ कर सकते हैं। और किस लिए? टू एनालाइज एंड इंटरप्रिटेशन ऑफ बायोलॉजिकल डेटा, कि उसके ज़रिए हम एनालिसिस करें और उसके ज़रिए हम इंटरप्रिटेशन करें ऑफ बायोलॉजिकल डेटा, कि इस यानी हम एनालिसिस करें कि ये डिजीज़ इस जीन्स की वजह से है, या अगर हम इसमें म्यूटेशन करें तो इसके ये रिजल्ट आ सकते हैं। और ये हम किस तरह कर सकते हैं? यूजिंग कंप्यूटर बेस्ड सॉफ्टवेयर एंड टूल। इसमें हम कंप्यूटर बेस्ड सॉफ्टवेयर और टूल्स यूज़ करते हैं। सो वी विल डिस्कस दैट व्हाट इज स्टोर, व्हाट इज रिट्रीव एंड व्हाट इज एनालाइज एट स्टेप बाय स्टेप। अब डेटा स्टोरेज, व्हाट इज डेटा स्टोरेज क्या है? डेटा स्टोरेज इसके मुख्तलिफ आप कह सकते हैं इसके मुख्तलिफ फॉर्म में हम डेटा को स्टोर कर सकते हैं। इसमें डीएनए सीक्वेंसेस है, आरएनए है, प्रोटीन सीक्वेंसेस है, मुख्तलिफ पाथवेज है, उसमें हम डेटा को सेव कर सकते हैं। नाउ डेटा रिट्रीवल। सो वंस द स्टोर डेटा, बायोइन्फॉर्मेटिक्स tools enable easy retrieval of specific information from these databases, allowing scientists to access the data they need for their research quickly. अब डेटा रिट्रीवल क्या है? अब हम फॉर एग्जांपल अगर हमें किसी जीन्स की ज़रूरत हो, एक स्पेसिफिक जीन्स पे हम रिसर्च करना चाहते हैं और हमें उसकी सीक्वेंस या उसकी क्वालिटीज़ की ज़रूरत हो, तो हम किस लिए हम एक डेटाबेस में जाते हैं। फॉर एग्जांपल हम NCBI में जाते हैं, वहाँ पे हम जीन्स का नाम एंटर करते हैं और वहाँ पे जितने भी इंफॉर्मेशन है वहाँ पे वो इंफॉर्मेशन हम रिट्रीव कर सकते हैं। अब ये किस तरह हम रिट्रीव कर सकते हैं? तो डिफरेंट टेक्निक्स होते हैं, डिफरेंट उसके लिए सॉफ्टवेयर्स होते हैं, उसके ज़रिए हम इसको रिट्रीव कर सकते हैं। अब अगर फॉर एग्जांपल अगर मैं कोई रिसर्च करो और रिसर्च के बाद मैं उनको डेटाबेस में सबमिट करो, वहाँ पे मैं उसको स्टोर कर सकूँ, तो वो पब्लिकली अवेलेबल होते हैं। तो दूसरा जो किसी भी कंट्री में हो, जहाँ से भी हो, वो उसको यूज़ कर सकता है उस इंफॉर्मेशंस को। Then data analysis. We will come to the third one, the data analysis. This one. So data analysis. This is the core function of bioinformatics. It is the main function of bioinformatics to analyze biological data using advanced algorithms, softwares, and computational techniques. This analysis uncover pattern, identifies gene functions, and help interpret complex biological phenomena and leading to some scientific discoveries. अब अगर हम फॉर एग्जांपल if we do some analysis and we want to check whether the other people also did such type of analysis and we want our result to compare our result with them, तो हम इसके लिए बायोइन्फॉर्मेटिक्स को यूज़ कर सकते हैं। And by this way we can compare our data with the previously published or the previously deposited data in the databases, and it will lead to the significant or the scientific discoveries. इसके ज़रिए हम फिर डिस्कवरीज कर सकते हैं, क्योंकि आपको एक चीज़ को आप अगर आपके पास डेटाबेस हो, अगर आपके पास वहाँ पे मटेरियल हो कि आप उसके साथ कंपैरिजन कर सके, तो इससे हमें वो एक ट्रेंड का पता चलता है कि किस तरह हम अपनी इंफॉर्मेशन, अपने डेटा को दूसरों के साथ कंपेयर कर सकते हैं और एक यानी कुछ कंक्लूड कर सकते हैं, और वही कंक्लूडेड जो इंफॉर्मेशन है वही जो हम फ्यूचर के लिए यूज़ कर सकते हैं। Before going to discuss the bioinformatics, first of all, we need and it is very crucial to understand something fundamental to biology. हमें उससे पहले एक इम्पोर्टेंट चीज़ को डिस्कस जो भी हम जब भी हम बायोइन्फॉर्मेटिक्स को डिस्कस करते हैं तो उससे पहले हमें एक चीज़ को डिस्कस करना है विच इज़ कॉल्ड द सेंट्रल डोगमा कि सेंट्रल डोगमा क्या है इट इज़ यू कैन से द ब्रेन इट इज़ द ब्रेन ऑफ मोलिकुलर बायोलॉजी. So now what is central dogma? The central dogma explain how genetic information flow from DNA to RNA and from RNA to protein. Simply in other words, these like if you say these are like DNA, RNA and protein. These are three biological molecules. Okay, they store, transfer and execute genetic information which is necessary for life. हर ऑर्गेनिज्म में ये हो पाया जाता है डीएनए, आरएनए एंड प्रोटीन. कुछ वायरस में ओनली आरएनए पाया जाता है. तो इसको सेंट्रल डोगमा कहते हैं. सेंट्रल डोगमा किसको कहते हैं? जिसमें डीएनए ट्रांसक्रिप्शन होती है आरएनए को एंड देन आरएनए की ट्रांसलेशन होती है प्रोटीन को. Okay, this whole process is called central dogma. So now you can see that in this we need DNA, we need RNA and we need protein. So we do bioinformatics analysis using these data, DNA, RNA and protein. So if you know about the central dogma, that what is the central dogma and how it works, so then you can easily understand, you can easily use bioinformatics software and tools to integrate your result, to check your result. The central dogma of biology describes the way in which information flows among these three classes. Among those classes are DNA, RNA and protein. In central dogma, इसमें क्या होता है? The central dogma, the first is DNA replication, in which the DNA will be replicate to DNA. Okay, in which the DNA will replicate to DNA. DNA अपनी एक कॉपी बनाता है जिसको DNA replication कहते हैं. The second one will be transcription, in which the DNA will transcribe to RNA. Okay, single stranded RNA. इसमें डबल स्ट्रैंडेड डीएनए को सिंगल स्ट्रैंडेड आरएनए में ट्रांसक्राइब किया जाता है. एंड देन द थर्ड वन इज ट्रांसलेशन, इन विच द आरएनए विल ट्रांसलेट टू प्रोटीन, जिसमें आरएनए को प्रोटीन में यानी ट्रांसलेट किया जाता है. एंड दिस इज द होल सेंट्रल डोगमा. हम जो भी रिसर्च करते हैं वो इन्हीं के गिर्द यानी घूमता है. So whatever we are doing a research in molecular biology, so we target DNA, we target RNA, we target protein. Either we will check the gene expression or we will check the protein expression. So bioinformatics help us study these molecules and their interaction by using computational approaches. बायोइन्फॉर्मेटिक्स हमें हेल्प करता है कि इन तीन मॉलिक्यूल डीएनए, आरएनए एंड प्रोटीन के इनके इंटरेक्शन को चेक कर सके. किस तरह चेक कर सके? बाय यूजिंग कंप्यूटेशनल अप्रोचेस. वेदर इट इज डीएनए और वेदर इट इज प्रोटीन स्ट्रक्चर, बायोइन्फॉर्मेटिक्स प्रोवाइड द टूल्स वी नीड टू अनलॉक द सीक्रेट ऑफ लाइफ. अगर हमारा रिसर्च डीएनए से रिलेटेड हो, आरएनए से रिलेटेड हो या प्रोटीन से रिलेटेड हो, बायोइन्फॉर्मेटिक्स हमें वो टूल देता है, हमें वो चीज़ वो प्रोवाइड करता है जिसमें वो ताकत हो कि उन चीज़ों को यानी अनलॉक कर सके. जो इंफॉर्मेशन इनमें छुपे हुए होते हैं उनको ये अनकवर कर सके और हमें बता सके, हमें विजुअलाइज कर सके कि दीज आर द रिजल्ट. Okay, now the biology, like as I mentioned before in the first slide, that if you, for like to make it easy, you can divide the bioinformatics into bio and informatics. So bio mean the biology and informatics mean the information and technology. So if you want to describe the bio, in bioinformatics. It means the biology in bioinformatics. So what we can study by using bioinformatics approaches. So in every organism we have DNA, and then DNA will transcribe to RNA and then protein, and then the molecular network, and then the cell, and then physiology or diseases. So what we can do in DNA, RNA, and protein, bioinformatics help us and provide us the tools which can help us in sequence alignment, database similarity search, motif finding. We will discuss these each step one by one in our next classes, inshallah. Okay, in DNA, the bioinformatics help us gene finding, that the gene is present in particular organism or not. Comparative genomics, to compare one genome with other related genomes, and DNA methylation. It is another process we will discuss in some other day. I don't want to make you confused here. Then what the bioinformatics help us in RNA. So in RNA, it is differential expression, that how many gene express in particular you can say treatment. Okay, like, and then co-expression, and then you can say that ncRNA. This is another thing we don't want to discuss here. Okay, this is the type of RNA. Okay, non-coding RNA, and then the protein. So how we bioinformatics help us to protein to analyze protein, mass spectroprotein, mass specific protein identification. Like, you can see the sequence of particular protein, you can predict the structure of particular protein, and you can align this protein with other protein. Then the molecular network, that protein interaction network. So bioinformatics help us that how a protein interact with another protein. Okay, how a protein help in disease control, how a protein helps to fight with some type of stress in the plant and animals. Okay, and then how a metabolic pathway activated and deactivated. Okay, now bioinformatics, the cell. So bioinformatics, when we know all these information, then we create a virtual environment to the cell, which is called simulation. ओके इसमें हम आपने मॉलिक्यूलर डाइकिंग और सिमुलेशन का नाम सुना होगा. तो इसमें हम एक इन्वायरमेंट क्रिएट करते हैं जिसको हम सिमुलेशन कहते हैं, लाइक इन्वायरमेंट इनसाइड द बॉडी ऑफ एनिमल प्लांट. उसके ज़रिए हम देखते हैं कि अगर एक जीन में चेंजेस किए जा सके, अगर हम एक जीन में म्यूटेशन करें तो हमें किस तरह वो रिजल्ट जो उसका पेरेंटल जीन है उससे रिजल्ट किस तरह चेंज होंगे, हाउ इट कैन अफेक्ट द रिजल्ट. Okay, and then the population genetics are human genetics physiology. For example, अगर हम एक mutation कर सके, if we do a mutation in a gene, so how it will transcribe to RNA, how it will make a protein, different protein, how this protein will interact with other protein, and how it will it will act, it will work in a cell environment, and then it will affect the disease. Okay, a particular anything you can say that. So it in the biology and informatics, it will work from genotype. This is the genotype. Genotype means the genetic makeup of organism. It will go from genotype to phenotype. The phenotype means the appearance of an organism. That how the genetic makeup affect the phenotype of an organism. कि किस तरह अगर हम एक प्लांट के जीन में चेंजेस कर सके तो ये किस तरह प्लांट के लीफ कलर या लीफ जो उसका शेप है उसको किस तरह चेंज कर सकता है. अगर हम एक स्पेसिफिक जीन्स उसमें म्यूटेट करें अगर प्लांट में, तो ये किस तरह फ्लावर की कलर को चेंज कर सकता है. तो ये है जीनोटाइप टू फेनोटाइप. Okay, now we will go to the second part, the informatics in bioinformatics. तो इन्फॉर्मेटिक्स इसमें क्या होता है, इसमें डेटा टू डिस्कवरी होती है. अब इन द प्रीवियस वन वी गॉट द डेटा इन द फॉर्म ऑफ डीएनए, इन द फॉर्म ऑफ आरएनए, इन द फॉर्म ऑफ प्रोटीन. अब इसमें क्या होता है, हियर वी will submit this data to databases. We will compare this data with other databases. Okay, databases, there are thousands of databases. We will discuss in the next class, inshallah. So databases, ontologies, metadata, and then data computation. That how there are hundreds and thousands of algorithms which we can use to analyze our data. There are hundreds of software, thousands of software tools, web server, and data mining. Data mining means that if there are some data present in some specific database and you want to just dig out something from that data, if you want to check their data by using specific software, by using specific tool, and you want to extract something from their data, it is called data modeling. And the fourth one is modeling and simulation, as I mentioned before, predictive model system simulation. इसमें हम प्रिडिक्शन करते हैं कि हाउ इफ वी मेक सम चेंजेस, सो हाउ इट विल अफेक्ट द डेटा. So we have the informatics part. This involves managing in computation data, making sense of the enormous data set generated by biological experiment. We use technique like data mining, data modeling, and simulation to extract meaningful information. You heard the word machine learning. So machine learning algorithm we can use to predict how protein will fold, how the protein folding will take place, and how a certain gene mutation might cause disease. That how if we make some changes in a gene, a mutation in a gene, so how it will affect the organism. Either it will cause disease in the organism, it will either it will boost the immunity system of that particular organism. So what will be the result? So these things we can first simulate, and then we can do in practically in some organisms. So I hope you will understand what is bioinformatics. Now, if we go to the objective of bioinformatics, so all we discussed previously, these are all the objectives of bioinformatics. So you can say the main goal is to understand the function and relationship of various components of biological systems. Okay, so development of new algorithms and statistics for assessing the relationship among large set of biological data. But bioinformatics we are using to develop algorithm, to develop new tools, to develop new statistic models, to develop new things. Why? To analyze the data, to analyze the complicated biological data. This help in identifying genetic marker, understanding disease mechanism, and even developing new therapies. For example, application of bioinformatics tools for the analysis and interpretation of the data, various biological data, it is the main objective of this bioinformatics. The other thing, bioinformatics aims to bridge the gap between raw data and biological insight. For example, if we think that we sequence, for example, if we do the whole genome sequencing of a particular organism, of a plant, for example, we take a plant and we do the whole genome sequencing. So now the data is raw data. It is only A, T, G, C, like these nucleotide. Like they will not show anything to you. It will be raw data in the FASTA format, in the FASTA format. But how will you get information from these? You will annotate the data. You will search the data in different databases. You will compare the data with different databases. So then you can get information from these sequences. So bioinformatics help us in development of databases for an efficient storage, access and management of large body of various biological information. So in bioinformatics, we will use those, because in bioinformatics you can use data in different formats. You can submit data in different formats, and there are different databases which can store data in different formats, and they try to you can say that they try to make your data like they will squeeze your data. Okay, they will compress your data in those databases. Why? Because of the storage problem. So whenever you want to retrieve the data, you can retrieve the data efficiently with some bioinformatics tool, and then you can use those data for your biological experiment and for your biological comparison. Now, the next important thing is the component of bioinformatics. Okay, that what are the components of bioinformatics? So let's talk about the core component of bioinformatics. The core components of bioinformatics is data, databases, and data mining tool. These three are the main component of bioinformatics. Okay, so first we have data. Okay, biological data come in many forms like DNA sequences, protein structure, and gene expression level. Okay, then we have databases which store this data in a structured way so that it can be easily accessed and retrieved. Okay, as I mentioned before. And lastly, we have data mining tool. Now what are the data mining tools that help us to extract this data from databases to find some meaningful pattern, to find some meaningful result. For example, in bioinformatics, you might retrieve a DNA sequences from database like GeneBank and then analyze it using a tool like BLAST. We have we have done this before, and when in our previous video when we are generating a phylogenetic tree. So in a phylogenetic tree, what we did, we take a DNA sequence, we took a DNA sequence, and then we blast that DNA sequence in the gene NCBI blast algorithm, and then we check that this organism is similar to which organism. Okay, why? To identify the specific organism. So this is like the blast as a tool. We use the NCBI blast as a tool. It is a bioinformatics tool, and we use NCBI as a database. And what was the data? The data was DNA data. Okay, so DNA data mining tools, these are the software and algorithms that we use to explore, analyze, and retrieve data from these databases. Okay, so this is like you can see that this is the databases. These are the three main component, and inshallah in the next lecture we will discuss about the databases in detail, that what how many type of this databases, and how we can store data in those databases, and why these databases from each other. Okay, and today we will discuss only about the data. So bioinformatics data come in many types. As I mentioned before, we have nucleotide sequence, which represent DNA. Then we have RNA sequences. We have protein sequences. We have protein structure. We have metabolic pathways. We have gel pictures, and we have literature. Okay. Now, DNA sequences, what are the type? Like DNA sequences is not so simple, that so in DNA sequences we can divide DNA sequences into many types. Some data we can submit to the databases in raw form, it is called raw DNA sequences. Then some are we can submit to the databases in genomic signature tags. This is which is called GSTs. This is a specific format for specific experiment. Then cDNA sequences. That what are the cDNA sequences? We will discuss these one by one in detail. Then organelle genome for example, what are the organelle DNA sequences? It is mitochondrial DNA, it is chloroplast DNA. Okay, so these DNA sequences also have specific databases. And then the RNA sequences, and then the express sequence tags. Express sequence tag also, it is related to RNA, and we have some specific tags. We have some specific you can say the primer, like So this is also a type of DNA. We can submit to the databases, and we can retrieve from the databases. Okay, RNA sequences, we all know that when we do the transcriptomic analysis of plant, animal, and anything, so we can submit those sequences to NCBI, and we can retrieve from the NCBI. The organic genome, I already discussed, it is chloroplast and mitochondrial genome, and we can use this chloroplast and mitochondrial genome for phylogenetic study, for for gene comparison, etc. cDNA sequences. It is also, you can say that one is genomic DNA, and genomic DNA, you have both non-coding region and coding region, but in cDNA you have only the coding region. Genomic signature tag and express sequence tag, it is almost similar, but genomic is the whole genome. In express tags only we have the genes which are expressed. And the raw sequence, raw DNA sequence, this is also called the SRA data. This is the NCBI. This is in this data we only submit the raw reads, which is directly come from the sequencing machine, the Illumina machine, the ion storage machine, the nanopore. So whenever we use this raw data, we submit this raw data to the NCBI database. Now why we use this raw data? Because, for example, if we get some raw data, and we did some analysis on that raw data, and we publish an article, so before publishing the article, the NCBI or the reviewer or the journal, they will always ask you to submit your raw data, to submit your genome sequence to the NCBI. So when you submit these sequences to NCBI, they will alert you accession number, and you can use that accession number in your article, in your paper. So by this way, the other people will know about where your raw data submitted, and if the other group in some other country, they want to again check your result, they want to again interpret the data by their own pipeline, by their own software, by their own tools. So it is publicly available, and they can use your data for their analysis. Okay, so this was all about the nucleic acid. The protein sequences, we know about the protein sequences. Then the protein sequences, we can submit the protein sequences, we can submit the protein structure, we can submit the protein codes, everything to the databases. And then metabolic pathways. So whenever scientists discover some specific pathway in particular organisms, so then they submit those pathway to the databases. And databases, when you have some specific expression gene expression analysis, so you can compare your genes in those databases. Okay, then gel picture, and some of the analysis, like PCR, gene expression, and every analysis. So we always do gel electrophoresis. So when we did do the gel electrophoresis, then we have to submit those gel spectra to the picture to the databases. Why? Because this is all for the community, for the scientific community. This is all for the like from our side to the contribution to the humanity. So you can retract, you can compare your gel picture with other. And then the literature, what is literature? So whenever we are publishing paper, article, whenever we are publishing reports, so these are called literature. So these literature also submitted to NCBI, and now the NCBI have automatic system. So when you publish a paper, so the NCBI automatically added it to the PubMed and one of the databases, and then you can see, you can extract, you can retrieve that paper very easily from these databases. So inshallah, in the next lecture, we will analyze deeper into we will go deeper into the databases, and we will talk about what databases are, the different type of databases used in bioinformatics, and how they help us organize biological data. So That's it for today video on bioinformatics. I hope you now have better understanding of what is bioinformatics and how it integrates biology with computational tools to solve complex biological problem. And मुझे उम्मीद है कि इंशाल्लाह आज की क्लास आपको समझ आई होगी. दिस क्लास इज़ ये क्लास बी स्टूडेंट के लिए भी है. ये क्लास एम स्टूडेंट या जितनी भी स्टूडेंट और रिसर्च में इन्वॉल्व है उनके लिए है कि बायोइन्फॉर्मेटिक्स के ज़रिए हम क्या कर सकते हैं और बायोइन्फॉर्मेटिक्स एक्चुअली रियल में है क्या. तो क्योंकि बायोइन्फॉर्मेटिक्स केमिस्ट्री में भी हम इस्तेमाल कर सकते हैं, फिज़िक्स में भी इस्तेमाल कर सकते हैं, मैथमेटिक्स एंड एवरी फील्ड वी कैन यूज़ दिस इज़ इंटीग्रेटेड फील्ड. So at the last, don't forget to like and share and subscribe for more videos, and we will explore these topics in detail. So see you next time, inshallah.